Metastasis is a mechanism by which cancer cells migrate to a new location within the body. Metastasis is actually the leading cause of death in patients with cancer. Therefore, if it is possible to slow down the rate of cancer cell migration, it can serve as a novel strategy in combating cancer cell metastasis. Taffy, or thrombin-activatable fibrinolysis inhibitor, plays a prominent role in mediating a balance between the coagulation and fibrinolytic cascades. Taffy is activated through cleavage of an N-terminal activation domain. It is cleaved by either plasmin, thrombin, or thrombin in complex with its endothelial cofactor thrombomodulin. This is the most effective form of activation as it increases activation by over 1,000-fold. Thrombomodulin has also been shown to be expressed in cancer cells and is inversely proportional to its malignancy. This has sparked interest in the contribution Taffy has on cancer cell metastasis. Once Taffy is activated, it exerts a basic carboxypeptidase function where it is able to cleave carboxyl terminal lysine and arginine residues on protein and peptide substrates, including the plasminogen receptor. The mechanism of metastasis is heavily dependent on pericellular plasminogen activation. Therefore, as I mentioned earlier, if Taffy can cleave carboxyl terminal lysine residues on the plasminogen receptor, it could slow down the rate of metastasis. The only problem is that Taffy is highly intrinsically unstable, thereby undergoing a conformational change associated with its loss of enzymatic activity. Taffy is characterized by its intrinsic instability, as it has a half-life of 8 to 15 minutes at physiological temperature. Therefore, if I've constructed a Taffy mutant that is independent from physiological activators and is stabilized through these stabilizing mutations located within the dynamic flap region of Taffy. Once I've isolated protein, I will be conducting invasion and migration assays to assess the role of this Taffy A mutant on breast cancer cell metastasis. In addition to this, I will be conducting half-life assays to assess the role of Taffy A stability in a cancer tumor microenvironment relative to the wild-type and stable form of Taffy. The objective of my project is to determine the extent by which this Taffy 8 mutant can prevent breast cancer cell metastasis as it is constitutively activated and stabilized.